Good morning, everyone. Can you all hear me over the speakers and everything? How's everybody doing today? Take a moment and sign in with that session ID, if you will. I've got a few clicker slides. I didn't have time to do a ton of them. I was in Essentia at like 7:40 for an 8 o'clock ophthalmologist appointment, and I didn't see the doctor until like almost 9:30. So then I had to rush up here with my eyes all dilated and stuff. And so, anyways, I can hardly see anything right now. So yeah, if I'm pointing at stuff that doesn't make sense, that's fine. Okay. Oh yeah, and I've also probably got glaucoma too. So just yeah, the good news keeps on coming, right? So, but that's my problem. So you can pass foundations first and then worry about becoming ophthalmologist, right? All right. Okay, so today we're going to talk about glycogen metabolism. We'll talk about the fed state process of glycogen synthesis, and then the fasted state process of glycogenolysis or glycogen breakdown. Okay. We'll then talk about the regulation of the two processes, and this will be an extension of our discussion of insulin receptor signaling and glucagon receptor signaling from earlier in the week. And then we'll conclude with a discussion of some inborn errors of metabolism that affect glycogen storage or glycogen metabolism. These are collectively called glycogen storage diseases. There are a dozen or more different glycogen storage diseases, and they all have a GSD and then some number, and then they also have frequently the name of the person who discovered them or first characterized them. So you don't have to learn every glycogen storage disease. I've picked out a few of them that I think are representing kind of the different spectrum of how glycogen metabolism can impact different tissues at different times. So read your objectives carefully and focus on those. Okay. Sound good? Okay, so for our outline of what we're going to talk about again, glycogen synthesis first, then glycogen degradation, then regulation, then disorders of metabolism. And in the inborn errors of metabolism, we'll discuss two clinical cases, and I think I've given you the initial first description of these cases, right? Did you have a chance to look at them? Any impressions? Glycogen storage disease, something you think? Okay, so yeah, there's kind of two presentations, sudden death on exertion with no history, and then this fasting hypoglycemia. Does it look like a fatty acid oxidation disorder? No. No. Why do you say no?
Elevated ketones.
Elevated ketone bodies. Yeah, good. Okay, so we'll go over those at some detail after the conclusion of the class. Okay, so should we do a few clicker questions? And these are kind of, going from all over the place, not exactly related to what we talked about on Monday. Remember that every amino acid has a keto acid transamination partner. And the alpha ketoglutarate, and I can't see, I can't read how many people have responded, so I'm just gonna go. If you need more time, just tell me here. Okay, the alpha ketoglutarate and glutamate, that's like the universal amine donor and acceptor system within most cells. Okay, so keep that one in mind. This is a question, an old question, and this is something I'm never going to ask you on an exam. I don't think that you're ever going to encounter a problem like this in the clinic, certainly, but if you want to get the gold star, you could consider this. Okay, so succinyl-CoA, first, that's going to be a substrate for the thiokinase reaction, right? And that's going to generate a GTP. Then you've got the fumarate reaction, which is going to make is that an NAD? And then the malate dehydrogenase, that's going to make an FAD, I think. Is that right? So one of each, I think, is the best answer. Good. So that's shown here. And, again, not super high impact, so I'm not sure why I'm wasting all of our time on it. But I think, answering questions like that, it's a good way to review the TCA cycle. And, it's easier to review the TCA cycle by solving a problem like that than by just, memorizing the enzymes, right? Okay, so this one is, again, hate to be a time goblin by asking questions like this, but, it's Friday, so. Ooh, okay. I can see there's a handful of people who selected the Swedish apple tree. And there you go. I think that when I first started asking this question, it was right when Dr. Shaw did an ethanol metabolism lecture too. So it had some relevance at one point in our curriculum, but not anymore. Okay. Okay, so just in a cartoon fashion, which of those molecules would look like glycogen? Have you? We ready to go here? Okay, very good. I mean, I think, either B or C, these are basically both branched polymers of glucose, right? So this structure, the linear structure, this would be like a plant starch, amylose. Then these two, they're both looking like glycogen. Glycogen is a little bit more branched or has more branch density, maybe is a good way to say it. So glycogen looks more like this rather than amylopectin, which is another plant starch that has a branch structure that is chemically similar to glycogen. And then this would be an example of like cellulose, another polymer of glucose that has the beta bonds, or excuse me, that has bonds that we can't digest, so an indigestible starch. Okay, so best answer would be this one, glycogen. So, yeah, I think that I've just said all this. So these are your principal polysaccharides that are made solely of glucose. Now we have other polysaccharides that would include other sugars that we can get from diverse sources. But in terms of like the caloric content of the carbohydrates in our diet, really we're mainly eating a lot of this. And then when we eat animal tissue, the animal tissue is going to have glycogen in it. So when we eat meat, we're getting some glycogen from there. Okay, and then we eat a lot of this too, but this doesn't really contribute to the calorie balance of our diet because it's pooped out essentially chemically unchanged from when we eat it. Okay, so let's consider glycogen and how it's made and what we can do with it once it's made. So glycogen is just to the west of glycolysis on our Stanford map of human metabolism. And it intersects with glycolysis because it's using a glycolytic substrate. Okay, so glucose 6-phosphate when it enters into a cell, it can either be converted into pyruvate through glycolysis, or it can be converted into glycogen through glycogenogenesis. So the significance of this pathway is that it's intersecting with glycolysis. Another place that it's intersecting with is, let's see, now we talked about glycosylation reactions as a post-secondary modification of proteins, didn't we? The carbohydrate addition to amino acids. Okay, so the carbohydrates, when they're added by the glycosylases, they're using nucleotide conjugated monosaccharides, okay? And they ultimately derive from UDP glucose, or glucose conjugated to the uridine diphosphate nucleotide. So all of the substrates for the glycosyl transferases that are responsible for making the carbohydrate chains on proteins, carbohydrate chains are also added to phospholipids in a similar manner using these nucleotide conjugated monosaccharides, and those all come from or intersect with the glycogen metabolism. Okay. So the purpose of the glycogen metabolism pathway in the fed state, glycogen synthesis allows us to store excess carbohydrate. In the fasted state, glycogenolysase allows us to gain carbohydrate energy from stored carbohydrate energy. Okay, is that clear? All right, we'll elaborate on that. Well, that's why we're here for the next couple hours, right? Okay. Okay, so if we just split off the, just looking at a blow-up of the carbohydrate metabolism pathway, we've already gone from glucose to pyruvates through glycolysis, and then we've discussed the TCA cycle and electron transport chain. So now our focus is again on this diversion away from glycolysis into glycogen synthesis. And the key intermediates, I guess, that would link, or the key intermediates that link glycogen synthesis with glycolysis are glucose 1-phosphate, which is a product of glycogenolysis, and it's also a substrate for making UDP glucose. UDP glucose is the ultimate substrate for glycogen synthesis. Okay, so this is a figure from one of my go-to textbooks, the Nelson textbook of pediatrics. This is a pretty good comprehensive view of carbohydrate metabolism within a cell, and if you look at what type of transporters it has, you could probably deduce what type of a cell this is, right? So I won't go through all of the details of this. You know, essentially we've talked about this before. Now we're going to talk about the glycogen metabolism part that's on the top here. So maybe just put a star in this and set this aside and refer back to it as you're reviewing this material and some of the gluconeogenesis material as well. Okay, now this is representing a hepatocyte or a liver cell. And you can tell that because it has the GLUT2 transporters, and it also has glucose 6-phosphatase, this enzyme that can remove a phosphate from glucose 6-phosphate to make free glucose. Now that is a process that is really restricted only to the liver and in some circumstances kidney epithelial cells. And the presence of this glucose 6-phosphatase enzyme, this is reflective of the different functions of glycogen in the liver versus everything else. And when I'm talking about everything else, I'm going to use a skeletal muscle cell as a prototypical example. But really any other cell that does carbohydrate metabolism is going to have a similar regulation of their glycogen utilization. Okay, so think of the liver does one thing with glycogen, all the rest of the body does a slightly different thing with glycogen. Now, chemically, the glycogen is identical between the two organs. There's no difference in the basics of how it's made or what it looks like after it's made or how it's broken down. The difference is really in the different functions of carbohydrate metabolism in the liver versus carbohydrate metabolism in other tissues. So in other tissues, such as shown here, a skeletal muscle cell, glycogen is serving as an intracellular carbohydrate buffer. So what that means is glucose will enter into a skeletal muscle cell, and if the skeletal muscle cell is doing work, of course, it'll be oxidized to carbon dioxide to generate ATP. If there's excess carbohydrate, the muscle cell will use that glucose then to synthesize glycogen. In the fasted state, the reverse process will release glycogen, or excuse me, release free glucose 6-phosphate from glycogen. That glucose 6-phosphate remains within the muscle cell. Okay, so that glucose 6-phosphate that is derived from glycogen breakdown in the fasted state in a muscle cell, that's only for generating energy within that muscle cell. Okay, so you could think of, in terms of the carbon from glucose, when that enters into a muscle cell, that's like a one-way trip. It's going to stay in that muscle cell until it's oxidized down to carbon dioxide. So the liver has a very different or subtly different, I guess, depending on how you look at it. It has a different, the glycogen is serving a different function. There, the glycogen is serving as a whole body glucose buffer. Okay, so the liver, because it has this glucose 6-phosphatase enzyme, this enzyme that can dephosphorylate the product of glycogen breakdown, that allows it to produce glucose that can sustain glucose homeostasis in the blood. Okay, and it can provide glucose in a regulated fashion to sustain blood glucose levels within that narrow window that we discussed on Monday, even in situations where you haven't eaten for days and weeks. Okay, so glycogen in a muscle or any other tissue besides the liver, that's an intracellular glucose buffer, and within the liver there, then that's the whole body glucose buffer. Okay. All good on that kind of big concept? And that we can illustrate that concept by looking at the pathologies of the different glycogen storage diseases. So many of the glycogen storage diseases will affect either the liver or other tissues like skeletal muscle. There are a few where they're affecting both tissues. So with the glycogen storage disease, such as the von Gerke disease, for example, that affect only the liver, the main phenotype or the main symptom there is reflective of the purpose of glycogen in the liver. People with liver-specific problems with glycogen metabolism will present with a fasting hypoglycemia. Okay, because this is what the liver wants to do, or this is what your body wants to do with the liver in the fasted state, but the liver can't do that. Okay, now the glycogen storage diseases that are affecting the muscle, that can have kind of two broad phenotypes. It can manifest as exercise intolerance, or pain on strenuous exercise, reflecting the energy starvation of the muscle cells because it can't access this intracellular glycogen buffer. Another way that can manifest is with cardiac dysfunction, and that can lead to sudden death because glycogen can accumulate abnormally within the cells and disrupt the function of the different fibers that are trying to contract and make a heartbeat. Okay, so I don't want to get too far ahead of myself, but kind of think about the two different functions of glycogen in these two different tissue compartments, and then you can use that to look at the symptoms and deduce where a glycogen storage disorder may be. Did you have a question?
Yeah, I'm like a little confused by all the different words.
Okay.
I mean, there's a lot of words, but just a few words. Glycogenolysis, gluconeogenesis, glucogenesis. Are there, like, I'm kind of confused what's what.
Yeah. Okay, and I'll try
Not
to.
going into too much detail. I don't know if we're going to do that later.
I'll try to ease your confusion rather than build to it. But there are overlapping names for some of these processes. I'll try to be consistent, but I won't promise. Okay, so gluconeogenesis, that's going to be a subject for next week, and I might touch on that. But gluconeogenesis is referring to the liver making glucose from mainly amino acids, but also glycerol and lactate that come from outside the liver. Okay, so the liver is taking up stuff and turning it into glucose and then spitting the glucose out into the blood. That's gluconeogenesis. So don't worry about that for the moment. Glycogen synthesis is the same as glycogenogenesis. Okay. I think I'm going to use glycogen synthesis mostly, but I may use glycogenogenesis. Glycogenolysis is referring to glycogen breakdown, and I think I'm going to try to say glycogen breakdown, but I may also use glycogenolysis. Okay, so that's kind of 5 terms covering 3 things. So is that good? Okay. If there's any other like vocabulary stuff or terms that you don't understand, please let me know, because there's no need for simple stuff to be a barrier to understanding. Okay, let's see. I'm just kind of going by shapes and colors on my screen here. I'm sorry. Okay, so in terms of the endocrine regulation and we touched on this back on Monday. So insulin, that is going to promote the synthesis of glycogen and the storage of glucose, excess glucose as glycogen. Then glucagon and also the catecholamines, epinephrine and norepinephrine, those are going to promote the breakdown of glycogen and the release of glucose 6-phosphate in all tissues, and then the release of glucose in the liver. Okay, so in terms of these 2 processes of glycogen synthesis and glycogen breakdown, there's really only 4 enzymes that you have to worry about that are actually doing the work of building glycogen or breaking it down. There are some regulatory enzymes that get a little bit more complicated later in the class, but in terms of the synthesis, we have glycogen synthase and branching enzyme, and then glycogen breakdown or glycogenolysis, the glycogen phosphorylase, that's making glucose 1-phosphate from the glycogen polymer. Then the debranching enzyme, this has, it's doing the opposite of branching enzyme, as its name suggests. However, there are 2 distinct enzymatic activities that are associated with debranching enzyme, and they can be genetically distinct. That is, you can have a mutation in the enzyme that will affect one function without affecting the other function. Okay, so in considering glycogen synthesis, we have to look at the structure of the main monomer in the glycogen polymer of glucose. So glucose, yes?
Sorry, this is on the last slide.
Okay.
Going back to the last slide, but is glycogen phosphorylase and the debranching enzyme both inhibited by PKB or then protein phosphatase 1?
The main regulation is of the glycogen synthase and the glycogen phosphorylase enzyme. The branching and debranching enzymes, I guess I'm not sure whether they're regulated through reversible phosphorylation like the rest or not. Yeah. So, yeah, you're welcome. So yeah, the key regulated steps are the glycogen synthase and the glycogen phosphorylase. Okay, so one of the terms that we'll refer to when we're talking about glycogen synthesis is the reducing end and the non-reducing end of the glucose polymer. And, like with the protein, which is a polymer of amino acids, the different amino acid subunits have polarity, and that gives polarity to the whole polymer. So we have a similar concept with glycogen as well. The polarity is a little more subtle than the easy to understand polarity of an amino acid peptide backbone. You have to consider the chemistry of the different carbons at the different ends of the glucose molecule. And the easiest way to do that is to look at the linear Fisher projection rather than the cyclic projection. So the non-reducing end of the glucose is referring to the, let's see, do I have that? Yeah, so the number 6 carbon is the non-reducing end. This is an alcohol carbon. The number 1 carbon that's sharing the electrons with this oxygen, this is an aldehyde carbon, and it's considered the reducing end because it could be reduced to an alcohol. Okay, whereas this one is already reduced or already in the alcohol state. Now, technically you could reduce the alcohol down to a methyl carbon or but anyways, that's where that nomenclature comes from. And that reducing end versus non-reducing end, that provides a directionality for glycogen synthesis and a polarity for the ultimate big polymer. Okay, so with glycogen synthesis, the first step in glycogen synthesis is the formation of glucose 6-phosphate. So, this is going to be a common first step when any carbohydrate enters into a cell. It's going to be first phosphorylated by a kinase, and that will trap it within the cell. So glucose can go through a GLUT transporter, but glucose 6-phosphate can't. Okay. So this first step, this is recapitulating the first step in glycolysis. Right, so this glucose 6-phosphate, this has more metabolic fates than what we're showing here, of course. Okay, so the next step if this glucose 6-phosphate is going to be committed to the glycogen synthesis pathway, is an isomerization reaction that changes the phosphate from that free number 6 carbon to a more constrained, if you will, position on the number 1 carbon. So this makes glucose 1-phosphate. The phosphorylation reaction, this, as you could imagine, coupled to the highly exergonic reaction of the hydrolysis of ATP, this is irreversible, but the phosphoglucomutase, that reaction is freely reversible. So this step has to be reversed when we're in the fasted state and we're breaking down glycogen and trying to make glucose 6-phosphate. Okay, so anyways, let's stick with the glycogen synthesis for the moment. Glucose 1-phosphate then is a substrate for the generation of the nucleotide conjugated sugar UDP glucose. So a molecule of uridine triphosphate is reacts with the glucose 1-phosphate. Pyrophosphate leaves the reaction, and you're left with 2 phosphates, one from the UTP, one from the glucose 1-phosphate, linking the glucose molecule with the nucleotide portion. And then, like with all of these reactions, not shown, but there's a pyrophosphorylase that can cleave the pyrophosphates into 2 molecules of inorganic phosphate. And, if you want to count ATP equivalents, you'd have to count that in. Okay, so once we have the UDP glucose generated, UDP glucose will have multiple metabolic fates. So UDP glucose can be used again for these glycosylation reactions, or UDP glucose could be converted into UDP galactose or N-acetyl galactose or some other modified carbohydrate for a glycosylation reaction. But for the moment, let's just stay with the synthesis of glycogen. Okay, so the synthesis of glycogen depends on a protein called glycogenin. So there's a glycogenin gene that's transcribed and translated into a protein. Different serine amino acid residues on the glycogenin are then the substrates for the initial addition of carbohydrate. So the first carbohydrates are added at a 1 to 4 linkage. So the number 1 carbon, or the non-reducing end of, or excuse me, the reducing end of the glucose is bonded to the protein. Then additional glucose molecules are bonded to the non-reducing end through this 1,4 linkage. And then that continues until thousands or hundreds of thousands of monomers of glucose are added to the polymer. Okay, so to look at this reaction in a little bit more detail, we've got our UDP glucose. So our nucleotide, our base and the ribose sugar and the phosphate. So our nucleotide is here, and then this is our glucose molecule. So the glycogen synthase enzyme, this is going to break the bond between the UDP nucleotide and the glucose, and then make a new 1 to 4 bond to the existing chain of glycogen with the new molecule of glucose. So this then is recapitulating the polarity of the initial substrate. So you've got a non-reducing end over here, and now you have a new non-reducing end with the addition of the new glucose monomer in the same direction. Okay, so the synthesis of glycogen is always towards the number 4 carbon. Okay, so you're always making 1 to 4 linkages, and when glycogen synthase is adding glucose, it's adding it in that direction. Okay, now, as you may imagine with a polymer like this, there's a lot of degrees of freedom, because you can have rotation and bond angle changing at each point where those monomers of glucose are added to the glycogen. Okay, so when you make a big chain like this, it's going to get super wiggly, and it's going to get hard for glycogen synthase to hang on to the substrate and build new glycogen. And it's also going to get hard for glycogen phosphorylase to hang on to that substrate in that super wiggly, lots of degrees of freedom state, right? So this problem is resolved by branching enzyme. And branching enzyme, as its name suggests, it has, the way to summarize the enzymatic activity is as a 4,6 transferase enzyme. So what it's really doing, it's cleaving a 1 to 4 bond. So it has a 1 to 4 glycosidase activity, and then it's making a new 1 to 6 bond. And that introduces a branch in the polymer, and then you have, instead of one end to add new glucose onto. So as shown here, if we get about 11 glucose units into the chain, then branching enzyme is going back into the chain and cleaving a 1 to 4 bond, then moving this portion back a few glucose molecules and making a branch point with a 1 to 6 bond. Okay, so the 1 to 6 bond, that's taking the free waving carbon the number 6 carbon and making a bond to the same carbon here as a 1 to 6 bond. So then you can have a new number 4 carbon that you can add more glucose onto. Okay, so 1 site of synthesis and 2 sites of synthesis. Okay, so this then progresses. We have the glycogen protein core here. You get synthesis of the glucose chain, and then every time it gets about 11 glucose monomers away from a branch point, then branching enzyme makes a new 1 to 6 bond, and that makes doubles the sites for glycogen synthase or glycogen phosphorylase to act. Okay, what questions do you have on that? Yes?
So it adds it like linear addition for like 11 chains, and then it branches or like splits it and makes a branch point. Does it matter where it makes that branch point? Like on your diagram, it shows you like coming off the second one in line. Is it always the second one in line, or can it be kind of anywhere to make a branch?
I don't think it's that consistent. I'm trying to look at the figure from Marx, and yeah, I think there's some variability with it. So it's moving backwards, just say a few, and keep it simple. I think it's, it's more the concept of why it's doing it is more important. So yeah, what advantages does a branched polymer like glycogen have versus, say, if we were to store our excess carbohydrate as amylose, a linear chain? Why is this better?
More dense.
It's more dense. Yeah, I think that's true. Sorry, yeah, go ahead.
You could cleave more off simultaneously.
I think that's kind of one of the big advantages, is that when you have a big polymer like this, say you've got this big polymer in a cell and you need to access some energy quickly. Each one of these new ends represents a site where glycogen phosphorylase can act and release new glucose 6-phosphate. Okay, so that allows you to release glucose from glycogen very rapidly in the fasted state, and it also allows you to pack excess glucose onto glycogen very rapidly in the fed state. Okay, so there's that. There's also, this idea of density. Like, do you want things to be very dense in the cell? When things are very dense, they can become insoluble, and that's kind of not good. Like, we don't want things to precipitate out of cells. And this branch structure, another kind of one of the benefits of this is it's increasing the amount of OH at the surface of the big polymer, if you will. So that's allowing it to get pretty big but remain soluble. Okay, let's see here. Okay, so let's look at the Marx conception of glycogen synthesis for a moment, and I think this is going to, again, recapitulate what I've said largely. Okay, so you've got, and this is, similar to DNA synthesis. Like DNA synthesis, you can't make the first nucleotide, right? You have to start with a existing chain and then extend it. Same idea with glycogen. You have to have that existing glycogen core made on the glycogen molecule, and then additional glucose molecules are added to it. Okay, so the glycogen synthase enzyme, this is using UDP glucose, and it's cleaving off UDP as a leaving group to add a molecule of glucose onto the chain. And it can do it onto this end. It can also do it onto this end. There are the liver-specific isoforms of the glycogen synthesis and inborn errors of metabolism related to it that will have a different phenotype. And I think we'll cover those shortly. Okay, so the glycogen synthase then will act until it extends that chain to the limit of the degrees of freedom, I guess, if you will, of the chain length. Then the branching enzyme will come in and make a new branch point. In this case, it's showing it moving ahead of the chain. In some other depictions of it, it's moving back here, so I'm not sure exactly which is the consensus. But anyways, it's taking a long linear part and making a branch part that has two shorter parts. Okay. All right. So the key enzymes here are glycogen synthase and then the 4,6 transferase enzyme. In terms of inborn errors of metabolism, glycogen storage disease 0 is an absence of glycogen synthase. And the glycogen storage disease 4, or Anderson's disease, this is a deficiency of the transferase enzyme. They both have fasting hypoglycemia as kind of a hallmark symptom. They're a little bit different in that the glycogen synthase deficiency, they don't have hepatomegaly or an enlarged liver that's associated with it. They actually have a small liver if you palpate it or if you look at it on a radiograph, reflecting just a complete absence of the synthesis of glycogen. So for the other glycogen storage diseases that affect the liver, they tend to be associated with hepatomegaly or an enlarged liver because you get this either normal glycogen that is accumulating in too much abundance, or you get a chemically wrong glycogen, if you will, that corrupts the ability to do normal glycogen metabolism, and it builds up. Okay, so let's see. I'll talk about the specific pathologies towards the end of the lecture.
So for Anderson's disease, it's just you're not making?
For Anderson disease, you're making glycogen, but you can't make a branch point. Okay, so you're making amylose essentially, and the amylose, that's going to build up, and once it's there, you can't really do anything with it. Glycogen phosphorylase can't access it easily, so yeah, it's it's a problem similarly to a lack of glycogen synthase, but in the case of Anderson disease, there is glycogen there. Okay, so I think we had this discussion earlier about the advantages of having a branch polymer, so I'm going to just skip that. Okay, are there questions about fed state glycogen synthesis? Okay, so I'll switch to the fasted state process of glycogen breakdown. This is, I guess, the debranching enzyme is a little bit more complicated than branching enzyme, and I'll elaborate on that shortly. Okay, so the key enzyme, or the key way that we get energy from glycogen in the fasted state, is through the action of glycogen phosphorylase. Glycogen phosphorylase is using glycogen and inorganic phosphate as a substrate, and it's cleaving the 1,4 bond at the end of glycogen, and then adding phosphate to the number 1 carbon of the leaving glucose to make glucose 1-phosphate. Okay. The glucose 1-phosphate then, this can go through the phosphoglucomutase reaction to make glucose 6-phosphate. At that point, what it does is going to be dependent on what tissue it's in. If it's in the liver, then it's going to be dephosphorylated to glucose and exported out of the cell. If it's in the skeletal muscle cell, then that glucose 6-phosphate is going to be used for glycolysis. Okay, so the glycogen phosphorylase is taking a free end of glycogen and clipping off individual units of glucose 1-phosphate And it does that until it is constrained by being close to this branch point. Okay, if you could imagine, there's a big enzyme that's grabbing onto this substrate, and as it's getting closer to this branch point, it's kind of getting jammed up a little bit. Okay, so at that point, then we have the action of debranching enzyme. Debranching enzyme has two separable enzymatic activities. It has a 4,6 transferase activity. So, a 4,4 transferase activity. So it's cleaving this 1 to 4 bond and then making a new 1 to 4 bond here. Okay, so remember the blue to the red circle here, that's a 1 to 6 bond. And then everything else is a 1 to 4 bond. So the 4,4 transferase activity is, and I've got that here, is referring to taking this piece of the glycogen chain and moving it over here to make now a linear glycogen chain with a stump that is a single molecule of glucose in a 1 to 6 bond. Okay? This stump then, this is relieved by the second activity of the debranching enzyme, which is a 1,6 glycosidase activity. So the 1,6 glycosidase is going to break this bond, and it's different than the glycogen phosphorylase reaction in that the 1,6 glycosidase activity of the debranching enzyme, that's releasing free glucose, whereas the glycogen phosphorylase is making glucose 1-phosphate. Okay, so this, when we're doing glycogen breakdown, we're making a lot of glucose 1-phosphate, and then we're making 1 molecule of glucose for every branch point that is resolved by debranching enzyme. Does that make sense? And in order to use this glucose for fuel within the cell, it's going to have to be phosphorylated to glucose 6-phosphate by hexokinase. If this is a liver cell, then this glucose can just go out the GLUT transporter and contribute to the glucose homeostasis in the blood. Okay, and these two activities of the debranching enzyme, these are separable activities, so you can have a deficiency in the enzyme by losing the 1,6 glucosidase activity while retaining the 4,4 transferase activity. Okay, but it still is a problem. So if you retain the transferase activity but you don't have the glucosidase activity, then all of your glycogen is going to get stuck in this point, and glycogen phosphorylase can't read through this. Okay, so you get abundant glycogen. You have no problem with building glycogen, but you can only break it down just to that one branch point, and then you're stuck. Okay, so we're getting kind of close to the end here. Let's see, so there are a couple of different pathologies that are associated with loss of function mutations in the glycogen degradation pathway. So glycogen phosphorylase deficiency in the muscle-specific isoform of that disease, that is McArdle's disease, and that can manifest as exercise intolerance from energy starvation of the tissue. HERS disease, this is caused by the liver-specific isoform or loss of function mutations in the liver-specific isoform of glycogen phosphorylase, and these will manifest as the fasting hypoglycemia and with an enlarged liver. Debranching enzyme deficiency, this is manifest as, this is referred to as Cori disease. There can be Cori disease or glycogen storage disease 3A and 3B, depending on whether the liver only is affected or whether it's affecting both skeletal muscle and the liver. Okay, and I'll touch on that later. What questions do you have?
So,
Go ahead. No, go ahead.
oh, so McArdle's is absence, and then hers is deficiency?
Same thing.
They're the same.
Yes, it's a loss of function mutation that you inherit from your parents. So it's present in all tissue from birth. Yeah, as opposed to like a mutation in one cell that becomes a cancer. So yeah, so these are inborn errors of metabolism, and the severe ones are going to be diagnosed in infancy generally. Well, I think that's safe to say. Did you have a question?
No, that answered it. So.
Okay. Okay, why don't we take a break and resume our discussion about the regulation of glycogenolysis and glycogen synthesis, and then discuss some of the inborn errors of metabolism.
The sixth one is not. Only one.
Like I'm gonna take my test and get 0 or
Same thing. So it's probably not as severe as these ones. This one comes.
No, nothing to do. We really have a limbo.
This sounds similar.
Yeah. And I think that
Corey's in the house.
even if you feel lysis, right, is like
Breaking down.
it's breaking down the storage so that it can be transformed.
Okay, so in order to make
So you are really
glycogen, you use carbon-coated bond between the end of one glycogen and a new molecule.
Discovering that as well.
I should. I'm not a good swimmer. It's really like a peaceful.
From that UTP, right? So those are high energy phosphate bonds. So it's a way of, if you could imagine like that carbon on the UDP glucose, it's by having it on the phosphate, it's like coiling the spring. So it's like giving it potential energy. The glycogen synthase enzyme can capture that energy and use it to make the new bond from the glucose molecule onto the end of
We
the
got on the ambulance, so it's kind of in my head for a while. But like I'm just like still heady, a little bit heady for me.
Well, but the branching so
Will also lead to a small liver because you cannot create.
So every one of these
So that's gonna be like amyloid.
ultimately coming from
It's gonna be hard for me to break it
Okay,
down.
so that's going to be the substrate for glycogen.
Of steric hindrance on that.
So the branching enzyme,
you just have a lot of.
that's not really using UDP.
Plus, you don't feel much
The
signal.
polymer that's already made, the chain of is already.
I'm just
So there's no phosphate
I don't know why I'm
involved in
like my
the branching enzyme.
How'd the other team do last time?
I think they won.
Whose team? So who's on your team? Like,
We are making.
That's
Yeah,
correct. Yeah, I mean, it's using these guys that once were UDP glucose, but that's from that part of it is gone.
Thank you.
And then we got some freshmen, who's our best player.
that's right. Good freshman.
What are you,
Our volleyball team.
oh, okay.
And then there's one more, who am I forgetting?
The other, and you said you don't know about the other team?
No, they won.
They did.
And did you guys kind of crush them, or it's pretty close?
we all know pretty tired.
Yeah.
Just the one, so it was I feel like
Got any pieces today?
You did?
Already? I wanna
Health, maybe? You play after four? Dude, good for you for just getting like on five.
Fractured your
Yeah, whatever that is.
polar aspect.
You're down.
Stag. I think it's stag. Yeah,
I can't even believe
Those are worse, dude.
they.
They're trash.
They weren't good.
No.
No, I can't.
There's no battery.
Yeah, yeah. Yes, yes, yes.
That's quite insane.